Hi everyone, this is Lecture 7, Cell Communication. So we have spent a lot of time doing the basics of chemistry and the components within a cell, and we are going to finish up this unit by talking about how cells communicate. So we're going to talk about the different types of cellular communication, how cells join together and interact when they are touching. Then we're going to talk about ligand-receptor type interactions and how, when cells are not actually touching, what types of chemical communication can happen. And then we're going to get into the specifics of the types of signaling that can happen through different ligand-receptor interactions. So whether cells can talk, whether the chemicals can signal from the outside of the cell and have to do a cascade within the cell, or whether the chemicals can get into the cell and make changes directly inside the cell. So, cell communication starts with the types. So each cell in the body is able to function as a single living unit. That's homeostasis, right? Every individual cell can maintain homeostasis, but the body as a whole functions because cells also communicate with each other. And that is how we get specialized groups of cells which have similar functions and will communicate to be specialized tissues. And that is also how we get specialized organs that can interact as a unit overall for the complete organ systems to maintain homeostasis in the body as a whole. So the types of cellular communication, we're going to talk about cell junctions, when cells are actually joined together. We're going to talk about cell-to-cell recognition. We're going to talk about the various types of chemical signals and the chemical signaling that can happen across short or long distances between cells. So first, this is how cells are joined. So cell junctions is when two cells are right next to each other, and there's different ways to join two cells. And these have to do really with the function of the tissues or the organ itself. So the first type is desmosomes. So these are when two cells need to be able to resist stretching. So these will be in tissues where there's a lot of pull or movement or elasticity of the tissue itself that is needed. And the desmosomes will anchor two cells together with some specialized molecules that we call cell adhesion molecules, or we abbreviate that with CAM, That prevents overstretching of tissues, and it keeps the cells very tightly linked together in tissues that are moving a lot. The next is tight junctions, and you will see tight junctions in cells of the body where we need to seal the tissues very tightly. So these are in areas where you don't want chemicals moving in between the cells, or you want to minimize that as much as possible. So tight junctions are very tight barriers that are going to prevent leakage between groups of cells. So they're going to form a tight barrier in a tissue group with the tight junctions. The last is the opposite of a tight junction. So the last is a gap junction, and a gap junction is actually a very porous hole. So you'll have two cells that are linked together by basically little tunnels. And there are tiny little tunnels, and the chemicals can move between the cells, and the cells basically then will act as a single unit. So instead of getting very, very large cells, there's sort of a limit on how big cells will get in organs. And if we then want to link the cells as a unit to get a functioning unit of an organ, we will then connect them by these little tunnels, and any signal that's in one cell will be able to move through that tunnel and get to the next cell. So the tunnels are limited, and there are specifics to that that you will learn about if you get into the different specialized cell-to-cell communication within each organ. So gap junctions allow direct communication between cells as they are basically sharing their cytoplasm and any signals that get into the cytoplasm itself. So here's a larger picture of the desmosomes where you can see the cell adhesion molecules really tightly linking those two cells together to prevent the stretching. And then here's a picture of the tight junctions. You don't need to worry about the kiss sites and the occludin proteins and everything that's labeled on here, but the idea here is that we have very, very tightly linked cell membranes from one cell to the next. And that's going to prevent any fluid from passing between cells. So that will create very tight barriers between these tissues. The last is gap junctions, and these are the little tunnels. See these tiny little purple tunnels here? So here's one side of a cell with a cell membrane, and here's another cell, and they are linked then by these little tunnels, which will allow some molecules to pass through and the cytoplasm to be shared. But it is limited, so there is no passage of very large molecules. So they can't share everything, but they can share things like ions, which would carry electrical charge, and other small signaling molecules. A little more specific on gap junctions, where the cytoplasm of the cells is directly linked by something called a connexon protein. So that's this protein here that's drawn in purple. And it allows ions and small water-soluble molecules to pass between the cells. Why water-soluble specific here? Well, because the lipids could pass anyway, right? So go back to your cell membrane transport lecture. Lipids would be able to get in and out of cell membranes freely. So these tunnels then, or the gap junctions, are allowing small water-soluble molecules to pass where they wouldn't normally be able to get through. It will cause multiple cells to work together as a single functional unit, and there will be synchronized activity between these cells. So a great example of gap junctions is in cardiac muscle. So you guys remember back to anatomy when you learned about the intercalated discs between two cardiac muscle cells. Those had gap junctions in them, allowing the heart to function electrically in a single unit or in collections of units, which we'll hear more about when we study the cardiovascular system. Okay. More on gap junctions, passing small molecules and ions through these connexon protein tunnels. Okay, so those are the way that cells are joined together. We also want to talk a little bit about how cells can recognize each other. So you can imagine if you want specialized tissues or specialized groups of cells, that there needs to be some recognition of similar and different, or self versus non-self. This is also important within the body, for the immune system to recognize what is part of the body and what is, for example, a bacteria or a foreign pathogen, which would be a non-self. So cell membranes, here's our membrane. If you look, you should remember this back from our membranes lecture. Here's our cell membrane with a lipid bilayer, and there are proteins there, which would help with many functions of the cell. And then here, these purple chains where you can see the sugar rings, these are carbohydrate chains. If the carbohydrates are attached to the phospholipid bilayer, then they're glycolipids. If the carbohydrates are attached to proteins, then they're that should say glycoproteins. I will fix that later for you guys for your PowerPoints. My apologies on that one. It should say glycoprotein. Actually, let's fix it right now. Easier to do it that way. Glycoprotein. Okay, there we go. So glycoproteins will be attached to the proteins. So this will help with self versus non-self recognition. So we'll have certain types of carbohydrates on your cells that are inside your body, and certain types of carbohydrates that will be on a non-self or a, say, bacterial cell, and then the body can recognize what should and should not be there. It also helps to mark boundaries of tissues, to mark cell tissue type, and prevent invasion of other tissues. So we will also segregate cells this way. So one way that the cells will recognize each other is that they will link up. So there will be receptors on one side that will link up these cell surface markers, if they are recognized, then the other cell will know, okay, that is a self cell. The immune system will do this. If it is not recognized, or it's recognized as something foreign, for example, a pathogen that we have been exposed to before, say E. coli or something like that, then the immune cell or that body cell will say, that's a non-self cell, and we will want to deal with that accordingly. Destroy it, mount an immune attack, etc. So that is cell-to-cell recognition. Now, when cells are not directly connecting or touching, we then need to use local and long-distance chemical signals. So in addition to having glycoproteins, in addition to having junctions, cells will also release chemicals, and cells can also receive chemicals from other cells. So chemicals will be secreted by one cell to another cell. And this usually requires what we call a ligand-receptor interaction, where the ligand is the chemical and the receptor is a protein that is on the other cell that will recognize that chemical. So let's talk a little bit more about ligands and receptors. So ligands, also pronounced ligands, I've heard it both ways, are extracellular chemicals. So it's really just another word for chemical. Okay, so a ligand is a chemical that it acts as a signaling molecule. So it's released by one cell to interact and talk to or send a message to another cell. So it's really a chemical message. Then, so here in this drawing, there's your ligand, right? It's this little yellow chemical here. It'll be floating around nearby, and then here's the cell membrane of another cell, which has a protein receptor. This receptor then has a specific part of it that will recognize the ligand when the ligand is nearby, recognizing that chemical signal and binds to that chemical signal, and it can make various changes. So the receptor is actually the protein itself that receives the chemical, and they are usually very specific. So it will only recognize one particular chemical or one small subset or group of chemicals that have similar structure. So receptors will recognize specific ligands or groups of ligands, and this binding is very specific. It's similar to the enzyme lock and key type binding. So many types of receptors can also recognize one ligand. So sometimes you could have a ligand affect one cell because it's bound to a particular type of receptor that will make the first type of changes in that cell. Or that same chemical on a different cell may interact with a different receptor that will make a whole different set of changes. So there are multiple possible receptors in many cases that can recognize one specific ligand, each having a different effect on a different subset of cells. So the possibilities here are huge. So ligand-receptor interactions are very general ways of chemicals interacting with other cells. There are many types of receptors. So, and we can't even get into all of the specifics here, but we're just going to do something very general. So there are receptors that are channels. There are receptors that are enzymes. There are receptors that are attached to a whole cascade of other proteins that can create sort of chemical domino effects in the cell. And there are receptors that are linked to the cytoskeleton that can actually alter the movement of a cell. Before we get into the types of receptors, let's talk about types of chemical signals, because this affects the types of receptors as well. So there are local chemical signals and there are long-distance chemical signals, and it's exactly what it sounds like. So a cell will send chemicals out to its target cell, the cell that it's trying to interact with and change. And that can be a cell that's very nearby, or it can be a cell that's really far away. And there's a couple ways of doing that. If they're local chemical signals, we have autocrine, paracrine, and neurotransmitter. And I'm going to go through each one of these in a separate slide. If they're long-distance chemical signals, we have hormones and neurohormones, and they're going long distance because they're going to travel through the blood to get to their target cell. Okay, so let's do paracrine and autocrine first. So paracrine signal is down here. So here you have a cell releasing a chemical, and it's going to affect a cell nearby. So in other words, it can just diffuse through the extracellular fluid nearby and get to a local target cell, something very nearby. It doesn't need to go through the blood or travel long distances. So paracrine is sort of a general local signal. Autocrine is an unusual case where the ligand is released and it affects itself. So that is auto for self. So this is a cell that releases a chemical and the receptor is actually on the cell itself. So it will change itself with its own signals. Some examples of these types of signals are histamines, cytokines, prostaglandins. You guys will hear some of these as we go through the different body systems. There's a larger picture of the paracrine signal, so the chemical being released from the secreting cell or the signaling cell goes through the extracellular fluid, diffuses, and then here's the protein receptor that's on the local target cell, very nearby. That chemical is recognized by the receptor, and depending on the type of receptor, it will make changes within that local target cell. The last specific local interaction is specific to the nervous system. And we'll talk a bit about neurotransmitters and a lot about electrical signaling in neurons when we get into the nervous system, which is our next unit. So neurotransmitters are secreted specifically from nervous system cells, which are neurons. So these are special cells that are specialized to have these synapses or synaptic junctions that connect to other cells. And they're very, very close. Very, very little extracellular fluid, just a small space between a neuron and its target cell. They are very local, they are very fast, and they are very short-lived signals. And we're going to spend a lot more time in the nervous system unit talking about neurotransmitters. But really a neurotransmitter is just another local signal. It's a chemical signal released from a cell to affect a target cell that has specific receptors for that chemical. So these will be specifically referred to as neurotransmitters, and these receptors will be referred to as neurotransmitter receptors. Okay, there it is again. And these target cells can be muscles, they can be other neurons, they can be glandular cells, lots of different types of synapses that you learned about in anatomy and that we will talk about again when we get to the nervous system. Okay, now for long-distance signals. So our long-distance signals have to go through the blood, and they're going to travel through the blood to distant targets, and we're going to separate them by whether they come from endocrine-type glands or nervous system cells. So if they are from endocrine glands, for example, the pituitary, the thyroid gland, the gonads, other endocrine glands, if they are coming from endocrine glands, then they are just general hormones. So these are chemicals that get into the blood and travel to long-distance target cells. Those target cells, in order to respond, have to have a receptor specific to that hormone. Neurohormones are very similar, but instead of an endocrine cell coming from a gland, these secretions are coming from neurons. So rather than calling these neurotransmitters coming from neurons, because they go into the blood, we're classifying them here as neurohormones. But you will see different textbooks just refer to these as neurotransmitters as well. Some people like to classify anything that comes from a neuron as a neurotransmitter. Here we're getting a little more specific because they go into the blood and affect long-distance targets. We're going to consider them to be another class of hormones or neurohormones. Again, just like the endocrine type hormones, the target cell has to have a receptor to respond to that chemical. Okay, here's a larger picture. So here's our gland cell, say thyroid gland, pituitary gland, etc., making its specific hormone, releasing that hormone into the blood, and then it's going to travel through the whole body. And it's going to specifically affect distant target cells that have receptors. Cells that don't have receptors will not respond to those hormones, and that's very important because we don't want non-target cells or the whole body to respond to a hormone that's specifically intended to make changes in only a certain subset or group of tissues. So those are general hormones, and then here's our neurohormones again. So there's our neuron secreting a chemical, which some people would just say is a neurotransmitter. But here, because it's not going to a synapse, it's going into the blood, it's going to travel through the blood, we consider it to be a neurohormone. And just like the other hormones, a receptor is required for the target cell to respond. No receptor, no effect on that target cell. Okay, what happens when these chemical signals actually bind to their receptors? So what happens when the ligand actually finds its receptor, or when the receptor finds its ligand? So the chemical signals of the ligands are going to bind to target cell receptors, and the response is going to depend. Isn't that such a science thing to say? Well, it depends. The response, but it is going to depend. We're giving you guys a very general idea here. But the response depends on the receptor type and on the molecules inside of the target cell. So this is how we can get one particular chemical signal getting very different effects inside different target cells. And you guys will see when we get to the endocrine system and we talk specifically about hormones, for example, how many different effects one single hormone can have on different types of cells. So these, if they're acting through ligand receptor, then most of the time they're acting through extracellular receptors. Some chemical signals, though, can actually enter the cell. They're lipid soluble. They can get through the plasma membrane. They'll get into the cell. They don't need a receptor, and they can actually affect the cell directly. When they do that, they can not only affect the molecules inside of the cell, proteins and other things inside of the cell, they can actually get directly into the nucleus, which has its own membrane, but again, that membrane is a lipid bilayer, and they can affect the DNA directly. They can also affect from that DNA transcription or protein synthesis and RNA. Some examples of cellular effects. So when these chemicals get into cells, they can change the activity of enzymes. So we spent a lot of time talking about metabolic enzymes or metabolism. So they can activate or inhibit enzyme activity and generally change metabolism of a cell, how active a cell is. They can increase or decrease protein synthesis, so they can change the secretions of a cell, they can change the chemical signals of that cell, other chemical signals. They can do that through activation of transcription factors directly on the DNA. They can also stimulate cell division, so they can affect mitosis and cell growth processes. They can stimulate it, they can inhibit it, they can make cells get bigger, they can make cells stop growing, many possibilities here. And finally, we'll do a lot of this when we get to the nervous system, particularly for neurotransmitters. They can alter electrical gradients. And this happens when you open and close specifically ion channels. And when you open and close ion channels, you will get changes in the charge, positive or negative ions, depending on what type of channels you open, and that can change the electrical activity within a cell. We'll get into a lot of that when we talk about nervous system signaling. So, how this happens, how these chemical signals make changes in target cells really depends on what type of signals they are. Are they lipid soluble, where they can get directly into the plasma membrane, or are they water soluble, where they'll be stopped by the plasma membrane and they need a receptor? So there are hydrophilic or water-soluble type hormones, and there are lipophilic or lipid-soluble type hormones. Water-soluble hormones are polar, meaning they have a charge. They will bind to receptors on the plasma membrane specifically because they can't get in the cell. Lipid-soluble are non-polar, or they do not have a charge, and they can enter the cell directly because they can pass through the lipid bilayer. When they do that, they will bind to receptors inside the cell and make changes directly inside the cell. So let's talk a little bit more. Is this super exciting? I know. Let's talk a little bit more about the classes of hormones. So water-soluble hormones, there are two types. Amine-based hormones, which are derived from amino acids. Some examples of those: epinephrine, thyroid hormone T3 and T4, serotonin, melatonin. Peptide hormones, which are small proteins. Some examples of peptide hormones are vasopressin and insulin. So these, because they're water-soluble, will be dissolved in the blood plasma, which is mostly water, and they will bind to extracellular plasma membrane receptors. So they're going to bind to the outside of the cell because they can't get inside the cell. Lipid-soluble hormones, for example, steroid hormones, which are made from cholesterol. Some of these are the ones that get a lot of press. So cortisol, estrogen, testosterone, aldosterone, which we'll learn about when we get more into the kidneys and blood pressure regulation, and then eicosanoid hormones. These are derived from fatty acids rather than cholesterol, and these are prostaglandins, leukotrienes, others. They cannot dissolve in the plasma of the blood because it's oil and water, right? Lipids don't dissolve in water. They will have to circulate bound to plasma proteins that are made in the liver. These plasma proteins, such as albumin, will carry the hormones, and then when they get to their targets, they will have to be released from the plasma proteins, and they will bind directly inside the cell and affect the DNA and related processes directly inside the cell. Okay, here's water-soluble hormones. And here's another picture here of how these work. So for water-soluble hormones, you're going to have your chemical or your ligand. It's going to have a charge such that it cannot pass through the lipid bilayer of the cell. So it will need to bind to an extracellular receptor, here's that here in purple, which is a protein, that has a specific binding site for that receptor. It will bind to that and make changes in that receptor. There are many changes and many possibilities here that we will get into. This one, for example, is an ion channel. So when this chemical binds to this receptor, it opens it up, and ions are allowed to pass into the cell. So here's the chemical binding, the receptor opens up, ions can now get into the cell. When the chemical is no longer bound, it will dissociate and the channel will close. So water-soluble hormones are extracellular membrane-bound receptor pathways. So they're going to be only signaling through extracellular receptors. The binding of the chemical, the hormone, or the ligand will cause a change in the receptor, and these receptors are proteins. Remember that the folding of proteins is very specific. We talked about primary, secondary, tertiary, quaternary folding of proteins. So what actually happens when these chemicals bind to the protein is it actually will change the shape of that receptor. Sometimes changing the shape of that receptor will open it up, like in the case of the ion channels, and ions can pass through. Sometimes it causes it just to bump into other molecules downstream. In either case, that conformational or shape change in the receptor because of the binding of the chemical will change something inside the cell. Usually, the activity of another set of proteins or group of molecules. The extracellular signal then has to pass through or is transduced, sort of translated, into an internal change in the cell. When the extracellular signal or the signal outside of the cell becomes a change inside of the cell, that transduction or that domino effect inside the cell we call signal transduction, because it's not the original signal that entered the cell. It's a second set of molecules inside the cell that had to be transduced. Lipid-soluble hormones, on the other hand, can pass right through the membrane. Look how easy that is. I'm sure the water-soluble hormones are jealous, right? So these lipid-soluble hormones can go right through the lipid bilayer, pass into the cell. They can also often pass directly into the nucleus and interact with some complex inside the nucleus. We call that the hormone receptor complex, or an intracellular receptor interaction, and that can cause changes directly to the DNA. If we change the DNA complex directly, then often that can change the activity of transcription and translation. We can get more mRNA, for example, and the cell can then make more proteins. This is a much more direct way to increase or change molecules within a cell. And often, it can increase or decrease protein synthesis directly. So these will bind to intracellular receptors in the nucleus to alter protein synthesis, but they have to be lipid soluble. The biggest class of lipid soluble hormones are the steroid hormones. Now, I keep saying we're going to get back to this, so here we are. Ultimately, the effect of a hormone or a ligand on a cell depends on the type of receptor that it activates. So here's some major types of receptors. Receptor ion channel is what I showed you already, where the receptor change opens an ion channel. We can also have receptor enzymes, Gproteincoupled receptors, and intracellular receptors. Let's look at those. Okay, we'll start with receptor ion channel. So here's your ligand, your chemical signal neurotransmitter, right? We've given it tons of names at this point. It's going to bind to the receptor on the plasma membrane. It will then make a change in that receptor. In the case of an ion channel, it opens the ion channel. Ions can move in, and we will get change in the electrical signaling of a cell. So in this case, the receptor actually is an ion channel. The ligand, oh, here's one more word for it, the extracellular messenger, binds to the receptor. Remember, these are often water-soluble because they cannot get into the cell. They have to have a receptor. So the receptor will change shape. That will open to allow specific ions in. When the ions get into the cell, that will cause a change in the electrical activity of the cell. Receptor enzymes are slightly different. So rather than opening when they bind to their ligand, they actually have changes on the internal surface of their cell. So in this case, the receptor has an enzyme attached to its internal surface. And when the chemical binds to that receptor, it alters the shape or the activity of that enzyme, and that will cause a change in some enzyme process within the cell. The enzyme then will become active and can catalyze or increase the activity of specific chemical reactions. So this is a way to get many changes within a cell in chemical reactions. And this can be metabolism, this can be a whole host of chemical reactions that can happen inside a cell. The downstream molecules then will become activated, changing activity within the cell. Okay, one example are tyrosine kinase receptors. So, for example, a tyrosine kinase enzyme has lots of phosphate bonds. So if you think back to your metabolism lecture, phosphate bonds hold energy. So we can use these phosphate bonds to activate downstream molecules. We can also use them to activate and create ATP. Or we can use them to activate downstream proteins. So a protein kinase enzyme will often take the phosphates from an ATP, hold on to them, then we can use these phosphate bonds to activate downstream proteins. Those downstream proteins could be other enzymes, they could be other receptors, they could be other molecules, a whole range of possibilities here that ultimately will change activity inside the cell. Okay, the next class is G-protein-coupled receptors. So G-protein-coupled receptors have this special sort of 1, 2, 3, 4, 5, 6, 7. They always have this little 7 transmembrane span. They kind of have this sort of snake-like look to them. They're interesting receptors that are attached to something called G proteins. These G proteins are activating proteins as well, kind of like the enzyme related or the tyrosine kinase. But these G proteins are a specific group of proteins that will activate another set of chemical reactions. So here you've got your chemical binding to the G protein receptor. We often abbreviate these as GPCRs, G protein coupled receptor. That will activate a G protein, which will then activate a bunch of other things downstream. Ultimately, it creates this cascade of changes, and because it goes through several other molecules, and in particular, some special molecules we call second messengers. We call these cascades. And these, because these second messenger molecules are important, we call them second messenger cascades. So this is where the receptor itself changes shape. That bumps or changes the shape of a G protein. That G protein then will activate a whole cascade of other proteins, and we call that a second messenger cascade. Second messenger cascades are very important in biology. They're going to come up a lot as you guys go through your clinical careers. So we need to get a little more specific about second messengers that can be used downstream of these G protein coupled receptors. So I'm going to show you 2 second messenger cascades. One is cyclic AMP. The other is the calcium calmodulin pathway. First, the cyclic AMP pathway. So we're going to draw this out and then we'll come back to the specifics. Okay, so for the cyclic AMP pathway, we are talking about, again, water-soluble ligands. So we're going to have a cell that has released a chemical. Often these are hormones that have traveled a long distance through the blood to get to their targets. The target is going to have a receptor that is going to be this interesting G protein coupled receptor on the membrane of that cell. So here's our target cell. Okay, here's our G protein coupled receptor that has recognized our chemical signal. This G protein coupled receptor has first a G protein attached to it. That G protein will activate the next molecule in line, which is the adenylate cyclase molecule. Adenylate cyclase will then produce or activate another molecule called cyclic AMP. I'll move this over a bit so you guys can see it. Cyclic AMP then will have many downstream effects on the cell, which can include increasing activity of the cell, growth of the cell, activating proteins of the cell, and we have a picture for you here, which we will add to the drawing. Okay, so let's do it both here and on the picture, okay? So you got your chemical attaching to your G-protein-coupled receptor. When the chemical binds, it's going to activate that G-protein. The G-protein will then activate adenylate cyclase, which is very close to the membrane. I should have drawn that a bit closer to the membrane. That is what we consider the effector, because that is what is directly changed by the G-protein. Adenylate cyclase then will produce this next molecule, cyclic AMP. Now this AMP is actually derived from ATP. The cyclic AMP then will have many downstream effects, and because it is the one that directly affects the proteins and other molecules inside the cell that we're interested in, we call the cyclic AMP the second messenger. And that is where these cascades get their name. So this is the second messenger cascade that will have many downstream effects on the cell. Okay, now let's look at this diagram, which shouldn't be as scary now that we've drawn it out. So here's our G-protein-coupled receptor attached to the G-protein, which will activate the G-protein, which will then activate adenylate cyclase, which will then make cyclic AMP. Many cyclic AMP molecules can be made and activate downstream effects in the cell. So for example, it can activate something called protein kinase A, and the kinase enzyme then is an enzyme that can phosphorylate or activate by adding a phosphate bond to many other proteins within the cell. So let's go back then to this general pathway. It is usually a water-soluble ligand that is activating an extracellular receptor. For second messenger pathways, it's a G-protein-coupled receptor that's attached to a G-protein that will activate adenylate cyclase, or AC. That will then activate cyclic AMP, make a ton of cyclic AMP for many downstream effects. One of those, for example, is to activate an enzyme. This enzyme, protein kinase A, can phosphorylate or add phosphate bonds to other cellular proteins. Those other cellular proteins then can make many other changes. For example, it could add a ton of receptors to the membrane. It could cause secretion in the cell. It could activate certain processes or enzymes within the cell. Ultimately, that causes many changes in the activity inside the cell. So that's the cyclic AMP second messenger pathway. I want you guys to know this one in great detail. So I want you to memorize that it's a G-protein coupled receptor through a G-protein, adenylate cyclase, cyclic AMP, and it can then activate many downstream effects. For example, protein kinase A, or PKA, which can activate other proteins and create a big response inside the cell. This one I'm going to give to you because you will also run into this in your clinical careers, but I don't need you guys to memorize this quite at the same detail, but let's do it. So again, this is a water-soluble ligand acting through an extracellular Gproteincoupled receptor. The effector here is not adenylate cyclase, it's phospholipase C. Phospholipase C will then convert something called PIP2 to something called IP3 and DAG. What that ultimately does is release calcium inside the cell. And the reason this is the calcium pathway rather than the cyclic AMP pathway is that calcium now is going to be our second messenger. And calcium will then have the downstream effects on the cell. It can form something called the calcium calmodulin complex, which can activate other enzymes downstream inside the cell, which will phosphorylate proteins. So you will see this a lot in molecular biology. You'll say, this molecule affects this molecule, which changes an enzyme, which phosphorylates proteins. And when we phosphorylate proteins, we create this big change inside the cell, which can be more secretions, more activity, more receptors. These are just very general pathways, guys. Here's a view of the calcium calmodulin pathway. Even though there's a couple of extra molecules in there, you'll see that it's very similar to the cyclic AMP pathway. So here's the chemical binding to the receptor, Gproteincoupled receptor. G protein is activated, which activates the effector. Here this is phospholipase C, which will then activate PIP2 to IP3. And then that will make calcium, or cause release, cause release of calcium. Calcium then will activate the calcium calmodulin complex, which can activate an enzyme which can phosphorylate downstream proteins. In addition to this PIP2 calcium calmodulin complex, the DAG portion of this interaction can also have its own activity as a second messenger. So DAG can activate a different type of enzyme called a protein kinase C enzyme, which can, guess what, phosphorylate a bunch of proteins and make a bunch of cellular activity changes. So if you want to sound really smart and somebody says, what does that enzyme do? You can say, well, it phosphorylates proteins, which causes a lot of changes in cellular activity. Very general, right? That's what these enzymes do. And it is the second messengers that will activate these enzymes. Ultimately, the point of this is signal amplification. So what we have is one single chemical binding to one receptor, which can cause literally thousands of molecular changes downstream through these second messenger cascades. So as that single chemical binds to that receptor, it turns on the switch. And that switch turns on these second messenger cascades as long as the chemical is bound. And we can create thousands of these enzymatic changes very quickly within the cell to create big activity changes within the cell through these second messenger cascades. So here's an example. One chemical can bind to one receptor, which can amplify and activate 10 adenylate cyclase. That will then multiply and amplify and activate 100 cyclic AMPs, which can then amplify and activate 1,000 protein kinase Cs. Then 1,000 protein kinase Cs can amplify and activate the enzymes downstream in the cell, phosphorylating different proteins. The products then of those enzymes can be in the millions. So one single molecule binding can create literally millions of activated molecules within that cell. Okay, so that was second messenger cascades. Now let's talk about intracellular receptors. So think back. What type of molecules can get inside the cell? You forgot already. No, you remembered. The molecules that can get inside the cell are lipid soluble. For example, steroid hormones. So a lipid soluble hormone will enter the cell and bind to a receptor that's inside the cell. So we're not bound to the membrane here in the case of our water soluble molecules. We can actually get in the cell directly. When it binds to its receptor, we then have a complex here, which our textbook is calling the hormone response element, or the HRE. So this is the chemical hormone binding to the receptor inside the cell, which creates the hormone receptor complex, or hormone response element. Then that complex can actually directly bind to, in many cases, directly change the DNA. That can alter transcription of a gene. Sometimes it turns it on, sometimes it turns it off. It really depends on the goal of that particular hormone and the pathway that's being used. In this case, if we activate transcription of a particular gene, then we will get more mRNA. More mRNA will then be translated into more protein. Those new proteins then can create a response inside the cell. So these can alter and change protein synthesis directly rather than phosphorylating proteins that are already there. So the second messenger cascades, the water-soluble type hormone signaling, generally changes proteins that are already present in the cell. The intracellular receptors, or the lipid-soluble molecules, can actually create and make new proteins within the cell. It's not that water-soluble molecules can't also do that, it's just it's more common with the intracellular type receptors and a more direct activation of genes here. That's it. So let me know if you guys have any questions. These are very general pathways that we are going to continue to use. So don't worry if this is all very brand new to you. We're going to spend a lot more time as we go through the individual body systems, going through and talking about the specifics, for example, in neurons, the specifics, for example, in different hormone pathways of how this cell-to-cell communication works. All right, let me know if you guys need anything, and I will see you soon.